With Puccinia graminis, for example, we have these different former species. There is evidence that in fact Puccinia graminis is not even a single species. Should we really continue to use the concept of former species? Some people are saying, forget it, let's just call it all Puccinia graminis and not bother. And I made the final comment before we broke for lunch that I think that there still is significant value in the idea of the concept of former species, particularly when we're talking to growers and when we're assessing the risk of rust. And to show you what I mean by that, I have an example here. If we talk about stem rust on barley, in Australia, if we find stem rust on barley, what we've found is, or what we know is, that it can be either, and in fact it's a little more complicated than this, but I'm just in general terms, it can be either the wheat stem rust pathogen, former species Tritici, or the rye stem rust pathogen, former species Cicalis, or it can be both. So if we find a crop of barley that's infected with stem rust, just by looking at it, we don't know whether the rust present is one of these or the other or both. Now that is important for a number of, working out which one it is important for a number of reasons, one of which is assessing the threat to wheat. So if we find a barley crop that is full of this one, we know that that rust does not pose any threat at all to people in the area that are growing wheat. So knowing which former species we're dealing with is quite important, and being able to actually tell growers in a particular area, you don't need to worry. Yes, we've found stem rust in barley, but wheat growers do not need to worry because in fact we've identified it as rye stem rust, not wheat stem rust. All right, so that's important from extension. Now to give you an example of that, I've been doing characterisation of rust isolates in Australia since 1988 is when I started. Stem rust in wheat and barley crops has been fairly rare, and in fact from 1980, particularly from 1980 through to 1992, it was very, very hard to find stem rust in a barley crop or in a wheat crop. But in early summer of 1992-93, we got a lot of rainfall. There was a lot of self-sown cereals growing, particularly in southeastern Australia, and this surprised a lot of people. Stem rust built up really, really quickly. So it came off a very low base. There was not much stem rust around. We went from a situation of having not much stem rust around to having a lot of stem rust in what we call the green bridge. These are the volunteer or the self-sown cereals that sometimes occur between the cropping cycles, and they're important in carrying the rust over from one season to the next. So what we found particularly was a lot of stem rust in self-sown barley, and a lot of the wheat growers were saying, well, what is the threat to us? We want to know what's there. Is this going to be a problem for us in the following growing cycle? I actually got a lot of isolates sent to me, and I found that 79%, let's say about 80% of those isolates from stem rusted barley were, I've got a typographical error there, I'm sorry, this word should not be there, were Puccinia graminis former species Cicalis, which does not infect wheat. So what we were able to tell the wheat growers is, yes, there is a threat, there is some wheat stem rust there, but the majority of that rust that's built up in barley is actually non-wheat infecting. It's former species Cicalis. And that was important information for the growers. All right, so yes, there was a threat, there was a risk, but the risk wasn't as great as what it would have been if all of the rust on barley had have been wheat attacking. Just a couple of final slides before we move on to pathotypes. So we also see these former species within the stripe rust pathogen, Puccinia striiformis. There's a form that infects wheat, a form that attacks barley. In Australia, we actually have a form that infects wild barley grass and really doesn't infect wheat at all, and very, very rarely infects barley. And then there's this one down here, which used to be called former species dactylus, that occurs on cocksfoot, or dactylus. But this recently was looked at by some Canadian people, and they actually consider it to be very, very different, and they've actually called it a different species altogether. So it's no longer known as Puccinia striformis, former species dactylidus. It's now known as Puccinia striformoides. Interestingly, this thing I think they've recently found, and Sajid, you might be able to comment on this. I think this has actually been found also on Barbary. Is that right?
Yes.
Yeah. So, I'm sorry?
Yeah. It's just that based on the sequencing, they have confirmed them to be different species, and then we have it on different species of Barbary. So.
Different species of Barbary to what you find wheat stripe.
Well, this is the raw data I have right now, but I have to look in further details. So.
Okay. Now that comment I just made about wild barley grass, this is another important thing for us in our extension in Australia. So we often see this wild barley grass growing as a weed in and around wheat crops, and we know that it's actually infected by wheat stripe rust, Puccinia graminis former species triticei. It's also infected by this Puccinia graminis former species pseudohordii, barley grass stripe rust. We know that the barley grass stripe rust poses almost no threat to barley and is not of any threat at all to wheat. So again, if we find stripe rust on barley grass, it's very important to us to know which one's there. We can say, yes, we've got stripe rust on barley grass, but the wheat growers want to know whether that's wheat stripe rust, wheat attacking stripe rust, or whether it's the barley grass form. All right, so again, that's another example of the importance of knowing which form you're actually dealing with. So just finally, I've already shown you that within Puccinia graminis, yes, we do have these specialised forms that occur on different hosts. In the stripe rust pathogen, we have these forms. What about leaf rust, the causal agent of leaf rust on wheat, Puccinia triticina? We don't really see the same kinds of former species in Puccinia triticina we do in Puccinia graminis and Puccinia striformis. We do actually see different leaf rust pathogens on wheat, but they're considered to be different species. Okay, so this is the one that we know most about, Puccinia triticina. It infects common wheat, and it can also infect some durum wheats. All right, it has the alternate host known as Thalictrum or meadow rue, which belongs to the family of the Ranunculaceae. There is actually another leaf rust that is far more restricted in its distribution. It occurs in Morocco and some parts of southern Europe, that infects predominantly durum. It's highly avirulent on common wheat, bread wheat, but it does actually infect durum. And this is called now, I believe, Puccinia recondita. And it has a completely different alternate host, Anchusa, which is in a different family altogether to Thalictrum. So with the leaf rusts that occur on cereals, we tend not to see these former species. But there are actually two different species of a leaf rust attacking pathogen that infect wheat, and they're discriminated broadly on their ability to infect durum. All right? Okay, I thought I might just, before I go to pathotypes, I thought I might just pick up on a conversation I had with Zach and I think possibly Gordon at the end of our last session before lunch. So you might be saying, well, what are these forms that we see within Puccinia graminis and Puccinia striformis, but we don't see them within Puccinia triticina? What are they? Where have they come from? Why are they there? Well, I think the reason is that what we're seeing is within Puccinia graminis and probably Puccinia striformis is what we call biogenic radiation. So we're seeing evolution that's giving rise to different variants that in time will become different species. Over hundreds, thousands of years through evolution, will probably become different species. What we're doing is we're seeing them at a snapshot in time, right? When they're on that pathway and they are actually diverging to become discrete species. So these things are not static. Evolution's going on all the time, but it's an incredibly slow process, and we're just seeing it at one point in time. So I think that's, to me, the reason that we do see these forms. They are actually diverging in their ecological niches and becoming separate biological entities, which over time will probably become different species in their own right. But when we're talking about that time, we're talking in terms of thousands of years. Okay? Any questions on that?
Yeah. How can one differentiate between Fuchsinia triticiana and Recondata? Is there any wheat line common on which one can differentiate, or Durham line?
So the question is, how do we discriminate between this pathogen and this one? To be honest, I've never worked with this. Well, I did a little bit of work with it in Europe, okay, but I'm not particularly familiar with its morphology. So I can't comment whether there are morphological differences or not. There may be, right? If there are, then clearly that's a very easy way of discriminating them. Clearly, the alternate host is very important in separating them, but that's a lot of work to actually go back to Telia and try and infect the alternate host to work out which one you've got. It's a huge amount of work. So generally speaking, the way of discriminating these two is their ability to infect durum wheat, right? So this one is highly virulent on durum wheat, but has poor virulence on common wheat. This one is sort of the other way around. It's far more virulent on common wheat. It can infect durum wheat, but it's not found very commonly on durum wheat. All right,
So if you had a few genotypes from each of those two different species, that would have a range of susceptibility, I suppose.
yeah,
You could separate them based on a...
yeah. So I do the race determinations for Puccinia triticina in Australia. All right, we don't have this one. Right, we don't have this one in Australia. So how would I know if that came into Australia? What would be the first thing that I would do? Well, I would take a sample and I would put it on a differential set, and what I'd probably see initially is that it's highly avirulent, and that would be unusual. I wouldn't expect that. So when I say highly avirulent, we'll talk more about this when we're talking about pathotypes, but what I mean is it probably would not infect many of those differentials. So straight away it would look unusual to me. The next thing I would do would be to take that isolate and test it on a range of durum wheats. Right, and if it was very virulent on the durum wheats, I'd be pretty convinced that this one's what I've got. If it showed the same level of virulence and avirulence on durum wheat as this one, then I would suspect it's just this one.
Yeah. Look, I just wanted to share with you the observation in India that we have. Race 77 is avirulent on durums. All the races which we have in India. But 104 group, there are two races, -1, -2, which go high on durums.
Mm-hmm.
But we used to call the whole thing as Puccinia recondita, but then later on it got changed to triticina. So we continue to call them P triticina races. But when in central zone, durum breeder wants us to send the brown rust, we send them 104 group, which is
Right.
virulent on
More virulent on durum wheat.
Yeah.
Yeah.
But we don't know whether recondita or...
Really, I think the only crucial way of knowing, absolutely, is to do the test on the alternate host.
To get hold of Benchusa.
Yeah. True. That's true. I think in time, again, once we get better molecular methods and sequencing, we'll probably have a DNA test that we can use for diagnostic, but at this point in time, the crucial test would be the specificity on the alternate host. And it could be the one, I would suspect the one that you're talking about, Prasha, is this one. Because we do know, and Dr. Colmer in the US has shown this. Dr. Colmer is a leaf rust expert, and he's actually shown that there are isolates of this that are adapted to durum wheat. So that makes the story a little bit more complicated.
Robert, I thought that recondita was the rye leaf rust.
Yeah, the name.
I thought Secalina was the rye stem rust, leaf rust, and I thought Recondita was now used for the durum rust, Zach, but I could be wrong. Do you know what the one that alternates on Anchusa is called?
No.
Yeah, we'll look into that.